Hello, everyone. It's a little bit after 10 and I've got quite a bit to cover, so I thought I'd just get right into it. So my name is Brant, and for those of you who saw the flyer and were wondering if that was me, you're sorely disappointed. If only I looked like that. Anyway, so I'm here to talk to you today about a technology called nanoString. Anyone here familiar with nanoString? Used it at all? Few? Great. Anyone doing gene expression studies? Anyone not doing gene expression studies? Sorry. A few that are not? Okay. Well, this technology is really focused on gene expression, or what you might do with the reverse transcription PCR, RT-qPCR. But there's other, it's generally very interesting. It's a unique technology, so I'm going to try and cover the technology with the understanding that, technology talks are very boring. When you're here to talk about an instrument, it's very boring. This technology isn't boring, but I'm going to divert from it a bit. So I'm talking about some of the landmarks around the technology that are really interesting and important to know. So hopefully it will be useful regardless. Okay. So nanoString, it might seem very odd to you, you may not have heard of it, but it's actually widely used around Australia. Here's a map of where all the installs are. The take-home message is it used to be a real strange thing. It's like no other technology, by the way, you'll see that. That's really where it finds its place, because it should be, when you hear about it, and when I first heard about it, I thought, why isn't this is going to take over the world, right? Now, there are reasons why it hasn't, which are crazy reasons, but it's really that good a technology and it's that different, which makes it something to know about and something to think seriously about using. But the point here is there's enough of these instruments around the country now that you all have access to it. If you want to run a nanoString experiment, we can get you to a lab so you can run it. That's essentially the message. Okay, so this is the platform I'm talking about. It's called the nCounter. Just find where my mouse is. Yeah, this is the nCounter. Comes in two parts. One's a robot that does it does a few things. It does liquid handling. It does electrophoresis, or electrostretching in this case. It does mag bead cleanup. It does a bunch of things, right? And the other one is essentially a microscope that's dedicated to looking at individual molecules. That's what happens here. So what the system is it's a digital system. In other words, we're counting de novo molecules, like the actual molecules get counted one by one. So you get a digital yes/no result, like it really is a number coming out. It's enzyme-free, which makes it unique, and that makes it extremely robust. There's no such things as dropouts and inhibitors and things like that. This thing just if hybridisation is going to work, this is going to work for you. The precision is about 8 times what the PCR is, for example, and the simplicity, it'll blow your mind how simple it is to use. It's a multiplex technology, so you do multiple things in the one tube. All your controls, all your running controls, plus, minus, negative controls, positive controls, processing controls, everything's built in, including all of your housekeeping genes and all of your targets for any number you want, up to a maximum of 855. It's mostly used for gene expression studies, but it works very well for microRNA, and also DNA when it's looking at copy number variations. These are kind of arcane and really limited to human and mouse, but for RNA, it does everything. Okay, so this is where I'm going to divert and do something I don't normally do, which is around, Houston, we still have a problem, right? And the reason I'm doing this is because I sat through a seminar just a few days ago, about a week or so back, by Joe van der Sompel. Does anyone know the name? Yeah. So he's got hundreds of publications and he's set up 3 companies, and he's one of the godfathers of PCR and then actually digital PCR. There's a cadre of these people around the world. They're well known, and that's been their life's work, really. So he gave the seminar on digital PCR. I was very interested. It was a great seminar. Here's just a little bit of a background on him. He's University of Ghent, but I've basically told you what you need to know. But his main point in that seminar was, is there a reproducibility crisis? Has everyone heard of this, reproducibility crisis in science? Oh, good, there's a few nods. Thank goodness. So the interesting thing here is most people agree there is a reproducibility crisis, because all the pink and reds are basically, yeah, there is. The little bit that's blue is No, there isn't. So we can sort of safely assume there probably is, and I believe there is. And if you Google this, you'll find lots, right? So here's a publication on it, the Reproducibility Crisis. You can look up some data on it around, you know, where is this reproducibility coming from? Is it just irreproducible in green? Is it lab error, analytical error, or something else? And you know, at the end of the day, this can result in retraction, and that's like a really bad thing, right? If you get papers retracted. Okay, but the PCR, like that's cool. Everybody here use PCR at some point? You rely on it? Day-to-day sort of technology? Good. Well, I'm going to get into it with you now. Well, there's a lot here that you may or may not be aware of, but you'll find a lot of this. You know, here's a publication, again, from one of the luminaries in the field. So Steve Buston is one of these guys who has built his life around the PCR, and he's been very outspoken about getting things right. In other words, making the reaction work. So here's one of his publications. PCR is probably the most widely used and enabling technology in molecular research, right? Despite the tens of thousands of papers, the number of clinical tests, like practical things to come out of it, can be counted on one hand. And we continue to face a crisis of reproducibility. So many papers published that report results that are meaningless. His words, not mine, but so back to Joe van der Sompel and his talk. In his talk, he brought up the MICE guidelines. Everybody knows the MICE guidelines, a few nods. These are things that, it's a list. Here's the list of things. This is my slide for it. When you're looking at complicated data like real-time PCR, you've got to get all the details right. And when you come to publish, you're supposed to go through this list and check everything off to make sure that you did all the right controls and things. Turns out there's 57 things that are considered essential you do, and another 26 that are desirable. The reason Michael Pfaffel and Carl Witwer are underlined here is I was talking alongside them a couple of years back. Actually, it's probably about 4 years ago now. And I presented nanoString at the same meeting, and they were very silent when they heard about nanoString. But they said to me, you know, Brant, PCR does work. It's just you've got to do all the right things, all the right checks and balances. And the problem is nobody follows the MICE guidelines. That was their admission, right? It can work, but it's really hard when it comes to publication and getting results right. And if you're a sequencing person, sequencing is not immune either because it's built on the PCR, right? And if you dig into it, there's lots of publications around amplification bias. And here we've got library prep bias and batch effects. I'm not going to go into these because, I mean, enough said, right? But it gets worse. All of that was about the PCR, but we don't do that for gene expression. We do reverse transcription PCR because RNA doesn't amplify. We have to turn it into a complementary DNA, right? So what do we know about reverse transcriptases? Well, a lot of this enzyme kinetics is a big variable: efficiency, specificity, sensitivity, the buffers, PCR inhibition. mRNA priming and bias, you know, it depends how you do that. RNA concentration effects, this is a real thing. People use carrier RNA and things like that. It's complicated. Target specific effects around secondary and tertiary structures of the target molecules. And basically, I mean, I would summarize that every RT reaction you do creates a different pool of molecules for the PCR, which is why when you do a PCR reaction, an RT-PCR reaction especially, you do everything at once and then you run the experiment, right? And when you come back next week or next month or next year or on the other side of the earth, you have to do it all again. You can't add to that result set because things don't add up. With nanoString, that's not the case, but it kind of tells you something's pretty fishy here. So a couple of more references for RT-PCR in case, you know, don't take my word for it. So here's another one from, I don't know the people here, but there's lots of publications like this. I just wanted to give you some flavor. So some RT systems contain substantial amounts of PCR inhibitors. Great divergence in efficiency are observed among commercial systems. RT is an important step in a gene expression study and should be considered with infinite cautions. I love that. Okay, another one here, properties of reverse transcription reaction, experimental variation, is mainly due to the reverse transcription step. We recommend extensive optimization, right? So here's another publication that, talks a lot about that optimization that's necessary. This is a Steve Buston paper. So, he really dug into it. Summarizing it, results showed variability sufficiently large to call into question the validity of many published data that rely on it. Now, there was a lot of recommendations in this paper. This little section here was their idea of how to optimize for an RT enzyme. You had to buy 4 of them, and you had to do a whole lot of work. And nobody does that. Nobody does it. Okay, talking the talk but not walking the walk. This is a famous publication. RT-PCR is a paradigm for the lack of reproducibility in molecular research. We conclude the majority of published RT-qPCR data are likely to represent technical noise. And he gets pretty wound up here. The unwillingness to take remedial action in general and for RT-qPCR in particular means that much of taxpayers' money and donors' generosity continues to be wasted. Again, not my words. And this is the same thing, RT applied to sequencing. There are problems there as well. In this study, they looked at formalin fixed, paraffin embedded. Anybody know what that is? If you're in the clinical space, that's how biopsies are preserved in wax blocks. You have to use that material, right? But they compared that material to fresh frozen, and only 3 out of 11 of those matched samples made any sense. So you can see there's a lot of reproducibility issues with this technology, even though it's used everywhere. Yeah.
It's mostly 2-step RT-PCR.
So I think single step's probably worse. Yeah, because you've got to do everything together. You can't optimize it separately. Yeah. Yeah. Well, things have come a long way in 30 years, but not that far. I mean, there's still a big problem with this. So it's not to say it doesn't work. I think the point here is you have to work hard to make sure that it is working, and then so validate it like hell, and then by all means, use it. I'm just trying to give you an alternative that just takes a lot of this noise away, right? So nanoString addresses every issue. I've been through this now for years and years. I've been doing nanoString I introduced the technology to Australia in 2013, and with the nCounter system, and I've seen everything. I've been into labs where they say, Brant, the nanoString data doesn't match what we've published with PCR. That's happened several times. And every time, the news for them has been bad. The nanoString was always right. So the big thing about the nCounter system is there are no enzymes. There is no reverse transcription bias. There's no reverse transcription. There's no polymerase. And what that means is all the things that can affect an enzyme go away. There are no amplicons. We have de novo detection of the molecule. There's no amplification of that signal. So there's no carryover contamination. There's no positive, pressure pre-prep rooms and post-prep rooms, none of that. There's no sample splitting. And you know what that means? Because the PCR is at 1-plex, right? You typically divide up for every target, and that means precision pipetting. If you don't pipette with absolute precision, you add noise, you add error. We don't do that at all. Everything's in the one tube, no matter the plex. So no precision pipetting. This is bucket chemistry. Every components in the reaction are in excess, which is why it's just so robust. Another reason it's so robust. There are no technical replicates. So people look at this and they go, that's impossible, Brant. You got to do technical replicates, dude. But the reason you do it for the PCR is you expect something to go wrong, right? You expect a dropout or something. And if you run 3, that's the minimum. You hope that if 1 goes, the 2 others are okay, and then they'll agree and give you an idea of what the real result is. But you should run lots of technical replicates, but everyone runs 3 because it's just too onerous and expensive to do it any other way. But the reason it works here is because we're doing population sampling. We're counting de novo molecule events. It's basically like counting all the people in this building, and and then you wanted to ask a question: how many males versus females? Well, it's pretty easy. It's pretty easy, right? And if you take a reasonable sample, that's all you need. So taking more and more samples just will end up with the same result, maybe with slightly better precision. But population sampling is the reason you don't, because if you get enough molecules to count, you get a very good result. And it's also because everything's processed in parallel. If there's going to be a problem, everything will fail. There's no halfway. It's like a digital TV picture. It's either perfect or, you know, you can't see anything. No off-target calls. People don't believe this because everyone's used to spurious events, spurious amplification, bad priming events, whatever it might be. But we've never, ever seen anything off-target with this system. Never. And there's hundreds of thousands of these been run now. Sample versatility. I mean, you can use just raw lysates. You use total RNA. Typically, you can use very degraded samples. You might have to add a bit more material to get the number of molecules with an intact hybridization site. But that's all. You know, raw lysates, people optimize for raw lysates, FFP, doesn't matter. It's very robust with sample type. So, you know, back in the day, I remember, I'll give you my worst example. I used to work a lot with a PCR. I did my first experiments with a CETUS instrument at Oak Ridge National Labs in Tennessee with my first postdoc. That was like the first PCR machine. So I remember all the crazy things. And I remember my worst ever sample was fungal spores. I could not amplify from fungal spores. Don't know what it was. Plants are not much better. There's polyphenolics and everything. We don't care. As long as what's in your reaction doesn't interfere with hybridization, there's no crazy salt thing happening in there, then all you need is a hybridization to happen with this technology. So look, last point here was saves resources, time and money, less expensive per data point. And the plasticware savings, because you're doing everything in one hit, in one tube, can be enormous. You can be looking at a handful of tips versus thousands to run an experiment. I have examples. I won't show you those slides, but anyway. So how does it work after all this talk? What is this technology? What's the trick? The trick is you just hybridize. Now, this is the best way to get to a technology, I think, is through the chemistry. So that's what I'm going to do. So here we're looking at an RNA target, and we're looking at a hybridization site of 100 bases. Firstly, there's a capture probe, and then there's a reporter probe. They just hybridize. Now, they've got a few fancy bits stuck on them. You've got biotin at one end to make it stick to a glass slide in the end. And these repeat hooks here are about processing. But essentially, you can ignore those. They're just part of the cleanup process, whether it happens on the instrument. The reporter probe is the bit that we see. So it has these fluorescent spots attached. These spots are gigantic. Actually, this is not to scale. That's 100 bases. But these fluorescent spots span about 7 kb or something. They're big, right? They're big, bright Alexa fluid blobs. There's 6 of them, and they could be in any one of 4 colors. So the arrangement of those colors informs what the molecule that it's attached to, right? So under the microscope, we see these things. And in fact, this is an image of what you would actually see. This is a Regis. No one ever looks at this data. The machine sees this data, but this is a reconstructed image from the different colors and you can see the barcodes are literally there. So if I zoom in on one, it looks like that. If I clean it up into a cartoon, there's your barcode. And under the microscope, it just goes around and adds all those up. If I can't see a clean barcode, it throws it out. That's a random event. It's okay to throw that out. And it just counts. And so what you end up with is, I'll show you what you end up with. We'll get to that. But that's essentially all it is. Now, the way the technology is mostly used in Australia anyway is with off-the-shelf panels. So these are highly curated target sets. And you can see some examples here for cancer, for tumor signaling, immune profiling. You know, there's more cancer pathways, breast cancer, cancer progression, myeloid innate immunity, host response, metabolic pathways, immune exhaustion, autoimmune profiling, fibrosis, organ transplants, and dogs, and maybe one more set, but then you get the idea. The thing is, these are all big. So if we look at this top one, for neuroinflammation, 770 targets, and typically it'll tell you how many housekeeping genes. I haven't listed on that one. Crazy me. But typically, yeah, here's a good example. 750 cancer-related targets here, and 20 internal reference controls. That's housekeeping genes. Why are there so many? Because you never know which ones you need. There's no such thing as a housekeeping gene. I'm going to get to that. But if you put enough in, you'll find ones that behave as if they are, and that's what you want. So That's the way most people use the technology. They're big curated biology systems, biology scale panels. They run it. They get enough data to typically, I've seen people get multiple publications out of one run because the data is so rich and deep and so beautifully curated. But that's really what people mostly use. But in New Zealand, it's the opposite. People do smaller scale things. And I thought that might be more appropriate here because you're not doing large clinical type work here, I would imagine. And you might be using odd species. So there's another chemistry, targets exactly the same thing, probe A and probe B. But this time, probe A and probe B are just generic oligos. So this is just like the PCR. You know, forward and reverse primer. This is like your forward and reverse primer. There's this oligo A, and probe B. And to make it all work for NanoString, we have these overhangs with specific sequences on them. And that one on probe A is designed to link to the identifier. Okay, so there's a tag there, links to the barcode to identify that event. At the other end, we just link on basically a biotin, and we're back to a usable chemistry. So all this self-assembles. You set up a hybridization reaction, it all joins together. Now, the interesting thing is the probe A's and probes B, no matter the chemistry, they sit right next to one another. There's no ligation to join them together or anything. But when they're like that, a few things happen. It's stable. Believe it or not, it's stable. So this tripartite complex, or in this case, it's five things together. It's stable. The RNA targets, of course, can be all different lengths. But the hybridization is done overnight. And in that process, they automatically nick down and basically disappear down to a tiny length. So they're all processed about the same length by the next morning. That's one of the tricks. And DNA is invisible unless you make it visible. So it's really nice. You can put anything you want. It just sees the targets you want, just sees RNA. Okay, so this elements chemistry lets you look at anything. And we've seen some projects on some wild things over the years. So how does it work in your hands? Is it something you should be scared of or not? Well, this is about as simple as I've seen anything in this field. You have 5 minutes of work to do that first day to set up 12 tubes. One run is 12 tubes. Now, you can get more than 12 samples out of that. If I have time, I'll show you how that's done. It's done with two-dimensional multiplexing of barcodes. So you can actually run 96 samples in one run. That's called PlexSet. I probably won't have time to explain it, but that exists. It's very simple. 12 tubes, you simply add a master mix, 8 microliters, 5 microliters of sample, and 2 microliters of the capture probe. That's it. So you can do that in 5 minutes. And it's bucket chemistry. It's not precision pipetting. On the next day, you set up the nCounter, and that machine runs always the same. So there's no variation in that protocol, whether you're doing microRNA or DNA or anything. You just load up the consumables, put your strip of 12 tubes into the machine and hit run. So that makes life easy. And at the end, you transfer that into the analyzer, which is the microscope. You can open it, you can pause it if somebody's already in there counting, open it, add yours, tell it to run it, run it in a queue, close it because it can run up to 6 of those 6 experiments in there by being interrupted and running. So that can run 24 hours a day. And again, that typically takes about 3 hours. So, by the end of that next day, you're going to have a digital data set. So what do results look like? They're incredibly, incredibly boring, right? They're incredibly boring, but they're so good, right? So when I talk about this technology, everybody has the word assay in their head, and they think of the PCR when they think of an assay, which is one gene target or one control in one tube. But here, every assay in the NanoString world is a self-contained single tube experiment. And that experiment can be run and the same thing can be run at any other time in anywhere else in the world, and the data can be combined. This is a weird and unbelievably good thing, right? Because everything's in there. Every control is built in. All the genes of interest, the reference controls, all the plus minus controls, all the processing controls, everything's in there. So if you look at a results set, it looks kind of like this. It's just a bunch of numbers. It just goes into an Excel spreadsheet, which is what I've got here. And you can see there's a set of positive controls, 6 of those. That's a 4-fold dilution series. You can eyeball that dilution series. These numbers are molecule counts. When you don't see a decimal place, it's a raw result. When you start using statistics, you get decimal places on things. But you can see there that's a standard curve that shows you linearity. It shows you that the assay is quantitative. And then there's that's that first block. The next block is also ERCC alien sequences. So these positive negative controls, they don't occur in nature. There's 8 of these negative controls. They're very important. This number will vary because individual molecules are sticky. So we don't expect this to be zero. Whenever it is 0, we put a 1 in so that it doesn't interfere with mathematics. Dividing zeros and things doesn't work. So you always see at least 1 molecule in the background. You can take, you know, the standard deviation of that and 2 standard deviations above that mean, and you get a line in the sand saying any counts above that, you're 99.99% real. So the statistics are very, very simple. The next block here is housekeeping genes. I would use at least 4, but more is better. I'll get to that. And then after that, you know, the endogenous is the genes of interest. And that could be any number that just grows up and down depending on what you want to do. So that's an experiment. There's one of these columns here. And these can be, you know, obtained whenever, wherever, and then pulled into a new analysis, and experiments can be combined. Any overlapping genes in there will be considered a viable experiment, and off you run. Okay, data analysis woes. Okay, if you're dealing with a data set that looks like this, you've got to eyeball it. There's no easy way to get around doing lots of manual intervention to get off the ground. And you can see in the background what digital counts look like. Now, digital counts is very boring, but it's very nice compared to a complicated data set like that. And the challenges you face with qPCR or microarrays or RNA-seq is that there's all these processing steps, every one of which adds error. You know, you can see the list here, you know, quality, quantity issues, manual pipetting errors, primer choice, transcription efficiency, blah, 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 down here. And over here, you've got technique bias, manual pipetting errors, reverse transcription efficiency. And then ultimately, what happens is these errors add up. And when you're doing something like next-gen sequencing, there's really heavy bioinformatics support required. But you end up with these other methods, you end up with more challenges with data analysis than you do with nanoString. On that score, this has just come up the other day. You can scan this now if you want, because this is happening on can you guys read this over me? On the 30th, April 30, there's an online webinar to go through nCounter data analysis. So these things happen through the year. There's lots of videos on nanoString's website how to do the analysis. So it's easy, and nanoString actually give you the tools to do it. Although there's a lot of third-party tools you can use as well. Okay, so great, Brant. You've told me this is great. Here's a little favorite saying of mine, which is show me the evidence, right? I don't believe anything until I see the peer review. So, okay, I'll show you peer review. This is a very good paper. It's from AstraZeneca, so it's from Big Pharma. But don't be frightened. One thing Big Pharma are very serious about is their methods. They generally refuse to use anything else but nanoString. They'll use nanoString in preference to everything else. And the reason is they've torn this system apart. They've looked at how they can break it in every possible way, how long they can leave every step, how much they can abuse it. And they've come out with this paper, which is available, came out in 2015. It's still very relevant because nothing's really changed with the technology. We found the practical aspects of the nCounter favorable over any other technique. Minimal time needed, reduced margin of error, few handling steps. And down here, sensitivity, technical reproducibility, and robustness for FFP samples. So there's key advantages for them. So I mean, and we know, because a lot of people in Australia who run core facilities, they will run pharma experiments, or we see multiple labs where the money has come from pharma to buy the instrument. Here's another good example. This is a guy called Matt Gadiris. He had a great webinar up. I don't know if it's still up on the website, but it was great because he was a lab there was a company called MitoBridge, and they were looking at mitochondrial diseases, and they're a big PCR lab, big RT-PCR lab, and they did not believe nanoString. So they went through and they pressure tested everything and compared it, and it's really interesting. And here's a couple of points. There's a lot they did, but a couple of points here. Here they had a protein X, compound B triggers significant upregulation of a target protein X. And they could see this on Western blots, but the PCR experiments had so much noise, they couldn't see the biology. But when they went to nanoString, the variation was almost zero, and they could see their biology very clearly. In fact, they could find, this is where I say the precision is unparalleled, right? 15% changes in expression. And that's really the spec for nanoString, which is 8 times better than the PCR, because the PCR, it's doubling at every cycle. So 100% change is what you're looking at there. And this is one of their points here. Thus, all variation was due to the RT-PCR, not biological replicates. Many people blame biological variation when often this is clearly not the case. And another thing they did not believe the arguments about technical replicates, but they did it themselves. They said they ran everything. Replicates were excellent across all samples, independent of experiment day, and this data convinced the lab we no longer need to run tech reps. Now, here's another one which I really like, because this is a researcher in New Zealand. So Kirsten works for the Plant and Food Research Limited company over there. They do a lot of things it's agronomic, but she works on kiwi fruit. And she'd been running PCR assays for years and having an awful time. And we convinced her, and it took a long time. It took us months and months, I think maybe even 2 years, to convince her to try nanoString. And we set up everything for her, right? And then she ran it, and she had a revelation. And she calls this career-changing technology. In fact, I'm setting up a speaking tour with her in New Zealand in a couple of months. She got so deep into nanoString, she convinced Plant and Food to buy an instrument that's like $500,000. And now she runs it for people all over New Zealand, and she wants to go around telling people more people about it. This is some comments from her. It's the height. She's been publishing. Here's one she did end of 23, which is recent. And then she sent me one just at the end in December. She said, here's another one. She said, this is the highest impact factor journal I've published in, 6.2, which is pretty awesome for plant research as opposed to human medicine. So she's done the hard yards with PCR, and she's completely converted. Here's another one. Axel Heiser, chief scientist at Ag Research in Palmy in New Zealand, Palmerston North. So this guy is very influential, very top-notch scientist in the country. He said, here's his quotes: I expected my initial enthusiasm for nanoString would be short-lived, as it always is, because new technologies rarely work as promised. But in fact, nanoString has exceeded expectations. We treat RNA samples very harshly. For example, we speed vac dry and resuspend, but it's very hard to ruin an RNA sample for nanoString in our experience. Future veterinary diagnostics is multiplexing, and this is perfect for nanoString. He also opens his machine up for other people to use. He's a big proponent of it, and he's done all sorts of things: human blood cells, plant cells, mouse embryonic, sheep embryonic, cattle, lots of things. Okay, so what I'd say to you is you might be entrenched in what you're doing, and that's fine. Finish what you're doing. But before you publish, it's a good idea to validate. And the best orthogonal validation method is nanoString. It's maximally orthogonal compared to the others because there's no reverse transcription, no PCR, and the readout is digital. That makes it the biggest outlier for orthogonality. Or is orthogonality a word? But anyway, that's what it is. So I would do that. It's easy to do. It's easy to run a small experiment. Okay, so sample input normalisation. Does anyone struggle with that? Is this the thing you talk about? Is everybody comfortable, or want a quick rundown? Because it's important. And over the years, this was the single biggest question that came up. I used to do all the installs for the nCounter machines. And in the question time, there was great discussion and arguments about this. Just crazy. So I'll go through it now. There is no such thing as a housekeeping gene. All right. And here's some evidence from nanoString. And this is from digital data, which is very clean. High expressors, medium expressors, low expressors. And these colored bars are different tissues. And you can see, sometimes a housekeeping gene works between a couple of tissues. But if you start varying the tissue, that the expression of those genes is all over the place, even though they're supposed to be reference genes, right? So what you've got to do is use multiple genes that you think or you've pre-validated are good reference genes, and then make a decision about the ones to actually keep and use on the day in that particular experiment. There used to be that, people would say use a high expressor, a medium expressor, and a low expressor. That's balderdash. Don't use low expressors because they can be more variable because you're at the noise floor. High expressors just take up space, right, on the chip, and they don't add much value. So medium expressors are kind of ideal, but it doesn't really matter. Honestly, it doesn't really matter. The only reason they used to say that was to try and get some variation in the number of in the housekeeping genes. Now, here's an interesting thing. Two people can do the same assay and get very different results. And that comes down to the choice of reference gene more often than you think. So here, the choice of one reference gene versus another can give opposite results. So the lesson there is don't use one housekeeping gene. That is absolute no-no, because it's like having one replicate. You don't know if it worked or not. And basically, another publication here basically said use as many as you can. I'm trying to cover this quickly for you. More reference genes, the better. This is interesting. Okay, you will not believe this if you haven't seen it before. Critical appraisal of qPCR results in colorectal cancer. You'd think colorectal cancer would be pretty serious stuff. And these guys went and they assessed all qPCR colorectal cancer publications from 2006 to 2013 for the number of reference genes used and whether they had been validated. Validity of 97% of those publications could not be assessed. 92% used a single reference gene. So you know what? Throw them all away. And only 6% used more than two. 70% used just one of the three reference genes, beta-actin, GAPDH, and 18S. Why? Because 20 years ago, that's what they said to do, and they're still doing it. Nobody's thinking or looking or reading. 30% of published reports, 30% publications report whether the chosen, or in other words, only 13% reported whether they'd even bothered to validate their reference gene. So you see, it's kind of green fields out there for doing this properly, right? The way that there's a few examples of this algorithm that they work much the same way, but the one that I would pick would be geNorm. It's publicly available. You've probably heard of it. So this is an algorithm that works with multiple reference genes. Here's how I explain it. You've got a small sample, so it's a small block, a big sample, and that's the variation you're trying to normalize out, the amount of sample you put into the assay. You've picked 4 housekeeping genes, A, B, C, and D, and you want to know which ones are working and which ones are actually behaving as a housekeeping gene. So what you do is you basically ratio, say, A to all the others, then B to all the others, and C to all the others, and D to all the others, and you end up with these. This is simplistic, but you end up with these ratios. If the ratios stay consistent in another sample, great. But in this case, everything that involved C was an inconsistent ratio, and therefore C is not behaving. But if the others are behaving, as you know, if the proportions of those stay constant, then clearly they're expressing to the same level from one assay to the next, one treatment to the next. So they're the ones worth keeping. And at nSolver software here, which is software that nanoString give you to do analyses with, it's okay. It's pretty good actually, but there may be better things out there now. But nSolver will go through, run geNorm, and in this case, they give a list of all these genes as being ones that actually worked using geNorm. And it put them in order, and it told you which ones to discard. Now, the reason you keep 6 is they will all have errors going in different directions. But if you take 6 and normalize and average all 6, then you get a very good normalization factor out of that. That's pretty much the state of the art. Okay, so I've set you up now for this next bit, which is what I'm going to finish on this. Good, I've got a bit of time left. I'm sorry I have rushed. I wondered if I'd even get this far, to be honest, when I looked at my slides. But anyway, so this is the other thing I hear from people. But the PCR can detect one molecule. It's much more sensitive than nanoString will ever be. So I'm sticking with PCR. Not so much. Depends on the context. So this is a thing called limit of quantification. It's different to limit of detection. So limit of quantification for a PCR is not one molecule. You can't quantify one molecule. I'll show you one in a sec. There's multiple publications on this. This is just one that I picked out. But ultimately, they all point to a number of about 100 molecules. If you don't put 100 molecules into a PCR, it will not be quantitative because there's stochastic sampling error, right? So I'm going to assume that's what it is, 100. And I'm going to assume a worst case scenario. I'm going to assume that in a nanoString reaction, we need 1,500 molecules because a lot of, in the processing, it all gets trimmed way down and a little bit goes on the glass slide, right? But if 1,500 go in, there's no stochastic sampling error and the whole thing comes out very reliably. And that's a big number. It's probably more like 1,000, but it's up in that range, right? So straight away you'd say, oh, well, nanoString's hopeless. It's actually zeptomole sensitivity, okay? 2 zeptomoles. That's incredibly sensitive, but the PCR is only 100 molecules. You go, oh, I'm going to stick with PCR. But you need to run 3 replicates. So there's 300 molecules per gene, This is stochastic sampling error. Molecules tend to clump. If you go into and you pipette out of a vial, a small sample, and there's not enough molecules in there, you could be taking out randomly nothing, in this case, or 11, or 3, or 6. So you can get hundreds of % error if you don't have like 100 molecules going in, right? That's the stochastic sampling error, and that's why it doesn't work. So yes, you can amplify a single molecule if it's there in theory. I went to a water testing place once. I won't tell you where. I'm not naming any names. And I had a bit of an argument because they said, we run 60 cycles, sometimes 70 cycles. I said, no, come on. You only need to run 35 cycles and you're at the one copy number range. No. It's important we do. And sometimes we need it. And I said, what? So you're detecting less than one molecule? You're detecting less than one molecule? And they said, yes, we are. And I got kicked out, by the way, for being noisy, or, you know, I mean, answering back or something, I don't know. So anyway, here's an interesting thing. So if you have a couple of targets of interest, say a gene and maybe a contextual target that backs up that gene behavior, that target behavior, and you put in, say, 4 housekeeping genes, which is kind of what you want, you're at 6-plex already, aren't you? So 6-plex. Now, at 6-plex, you need 300 plus 300, you need 1,500 molecules going in there because you've got to take that sample and split it out all those different ways and deal with all the errors of that. Whereas that whole sample doesn't need to be split. You just put that 1,500 molecules into one vial and everything is added in a large amount, right? So at that point, there's your break-even point, really. So if you're doing 6-plex or more, nanoString is going to end up more sensitive. And by the sensitive, I mean, you know, you need less. Yeah. Do you require more or less sample? Less sample is more sensitivity. So here, one target, 1,500 molecules, PCR 300. But as this number goes up, you know, sorry, 5-plex here. Sorry, it's 5-plex, 5 times, yeah. So it's 1,500 molecules, 1,500 molecules. And as you go up in plex, you don't have to add any more sample with nanoString, but you have to keep adding a lot of sample and splitting it all those different ways to run PCR. And if you want to look at what that looks like as a graph, this is the nanoString input amount staying consistent, 1,500 molecules. And this is as your plex goes up, it gets ridiculous. Does that make sense? Okay, so I can end here. I have some more stuff I can add on the end depending on questions. So the nCounter, you go from total RNA to a result very, very quickly. It's overnight. Set up one afternoon, hybridize overnight. It's about 14 hours of hybridization is the norm. You can do longer. You can do, the longer you hybridize, slightly better count. So you can hybridize for 3 days if you want to. It doesn't really matter. But typically 14 hours, everyone does. Works with the most difficult samples, which is great because some samples are really difficult if you're dealing with, I don't know what it's like working with marine samples, but woody plants and fungi are really terrible. Plex is trivial, use as many genes as you want, many housekeeping genes as you want. And you just saw the data there for when you get to 5-plex or more, nanostring's more sensitive. I can break there. I can add more. It depends on if there's any questions and how you want that. But yeah, please. I can repeat the question. Oh, good. There we are.
Yeah, I guess my background got a bit of experience in it, and I reckon it's probably 2013. You said you kind of sold it to Australia, and I was like, oh, hooked on it. Great, no
Yep.
problem.
Early days. Yep.
Yeah, I guess since then we've only really used it once. But I guess what people want to know is, the process of, I guess, numbers, like,
Costs?
at that point it was kind of, and maybe it is now,
Yeah.
if you've only got 10 genes, do we use our digital PCR here or
Yep.
qPCR? For me, it was,
Yep.
if you've got 100, this makes sense.
Yep.
But you've also got to be aware that it's going to be something like $20,000. And we're not big pharma.
Yep.
We might have limited cash, but
Yep.
yeah, so that kind of thing, but also knowing that we're not going to do it. We don't have a machine here, but it's somewhere here. What's the process from start to finish? Yeah.
Okay, no, they're really good questions. I kind of anticipated that actually. So where do I start? Are they, in fact, this is the real question. Can we afford to use this thing? I'm actually a fan of digital PCR. I mean, PCR, every technology has its sweet spot. Sometimes you want to know, like you really do want to know if you could find that one molecule, right? So you really do need that level of amplification and sensitivity. And that's where droplet digital is great. Well, digital, not the droplet thing you don't need anymore, but digital PCR is great. I think its challenges is it's a lot of messing around and the plexing becomes more of an issue, housekeeping genes, sort of thing. So it depends on what you want to do. It's not the cheapest technologies either. I can give you a reality check on what a small experiment would look like. In fact, this is how we get a lot of people started. If we use the elements chemistry, you would buy it. The smallest pack size is for 12-plex, so 12 barcodes and 1 run. And that will cost you $1,000, $1,070 with everything, except the oligos. Now, I've said that down to the upfront cost of PCR primers and nCounter probes is not included, because when you buy them, you're making a commitment to run, you could run tens of thousands of assays. I think it's a theoretical 1 million assays with the nCounter if you order the oligos. So it's a resource for the lab into the future, right? So I'm taking that out, because if you added that and you only ran one experiment, it wouldn't make sense. But these are the running costs. So this includes everything, including all the bioinformatics design. All designs are free, as you probably know from nanoString. They give you a really detailed report and they send you the ordering form, so you can go to IDT and just fill in your details and they'll send you what you ordered without any transcriptional errors of copying and pasting onto an order sheet. So it costs you $1,000. Remember, this is 12 targets and 12 samples. When you divide that down, it comes to $7.43 per result. If you were to do that with RT-PCR, you'd need to do each technical replicate for less than $2.47. And we know RT-PCR is more expensive than that. When people have compared true apples to apples, they end up in dollar ranges that are up around $10 to $20 here, not $2.47. And you can see if the plex goes up, the cost doesn't go up much, and the final cost per target comes way down. And the other thing you can do is, if you bought this and you had only a 9-plex, for example, not a 12-plex or a 7-plex, you can just not design for all the barcodes, and the barcodes that are unused are just ignored. So this will be the low end. This is the worst case scenario here. And even that's not looking too bad, I think. Remembering that you'll get the result very cleanly with a very, very high precision, and no one's going to argue with it. Like Kirsten Worm said, she went straight to publication in the top journals because no one questioned her. Her results were pristine. Whereas, you couldn't make sense of her other data. Does that kind of get to that? Now with the big panel products, you're looking at thousands of dollars an assay, and maybe you're crossing $8,000 to $10,000 sometimes. maybe it's not that much, but anyway, it's a significant amount. But again, when you look at it from a value point of view, a lot of people use it because if they've got difficult samples, maybe samples that have failed by sequencing, they will run with the nCounter. And often people come across because they've got some intractable problem, the samples don't work or whatever, or there's not enough of it. They can't split it many ways, so they keep it all together, put it into one tube, and the nanoString pops it out. Yeah,
that's kind of what I thought, but it's good for the explanation for everyone. And I apologize. I think it was Jess here. Yeah, we had it in the grant. It was the seaweed grant, and
right.
Yeah, I put it, and it was going to happen, but yeah, you're doing qPCR. Yeah, and it's seaweed, so it's like plants.
Yeah,
technically.
Seaweed's probably not that easy, right?
Yeah, it's got all these plant-related issues.
Yeah, look, I've been through all of that pain. I have, and I worked for Corbett for a while. You might remember Corbett made thermal cyclers. I was their global marketing manager and everything. That was the rotational system. So it took away thermal variation because it just rotated the block at high speed. That was pretty cool. But the PCR is the PCR. I had the number plate in Victoria when I worked in Victoria. I had the number plate PCR for Victoria. So I was really invested in that. I really was. I worked for Applied Biosystems and I released real-time PCR to Australia and New Zealand back in the day. I left academia to do that because I thought real-time PCR was so cool. But after 30 years, something better has got to come along, right? And when you understand this, when you get your head around it and you use it, and from your point of view, these machines are available. There's several in Queensland. The biggest and best facility in Queensland is at Griffith University on the Gold Coast. It's not far away. It's only less than 2 hours away if you drove it down. And Nick has 2 of these machines, right? He has everything. He's got more nanoString instruments than anyone in the Southern Hemisphere.
Yeah,
so,
and we've worked with him.
You work with Nick.
He's pretty quick, yeah.
He's better now. He's better now. He's grown, I think. Yep. Yeah.
Yeah, that's what I was going to say. There's a backlog, I guess.
Yeah.
Or he didn't reply to you.
There used to be. He's now got double he's got double the encounters, and he's got a whole different setup now. So yeah, in the early days he was very keen to run as a business, and now he's got big pharma, and it's quite a changed. But he did run the assays for you, and it probably wasn't as breezy as it is now. Is all I'd say. But he's very experienced. But
he doesn't put us smaller people in the back. Is it?
No, he
right?
does not.
The big pharma, yeah.
No, he does not. In fact, it's in his interest. He's incentivized to do these sort of projects for researchers. The university sees the money coming in from pharma, but they keep hassling him about doing and collaborating and this sort of thing because it's all about Australian research. Yeah.
Thanks. I'm sold. I think I unfortunately not going to use it because for lots of our applications, it's not. We're not doing it in high enough numbers of replications to justify the upfront cost for the probes, which I don't know, by the way, but I imagine that's.
I think they're around $60-something a pair.
Okay.
It's not too bad.
Not too bad.
And there's some tricks. You can't order them from anybody. You should order them from IDT. They found out in the early days, this is a trivial thing, but it's interesting, that if they made probe A's and probe B's in the same place, there'd be contamination and things would go funny. And after this is the early days of nanoString. Now they make them in completely different facilities and then send them to you because otherwise there's carryover from one synthesis to the next.
I'm curious, this water sampling facility, which jurisdiction is it? Is it in Australia? So I know not to drink off the tap there. No, that's outrageous, really. But
yeah. No, it's true. It's true. They told me they could collect, you heard the story.
Yeah,
That's true.
No, but seriously, I think, are you aware of lock nucleic acids chemistry? Because I think there's some synthesis that could happen in the nanoString if you apply that and do multiplexing of multiple different type molecules. So out of 12 potential probe pairs, you can amplify hundreds potential targets depending on the species and the bioinformatics in the background to understand the transcriptomics in that.
Is the question about
the chemistry of the probe itself.
Yes.
So there's locked nucleic acids.
Oh, yes.
Yeah.
LNAs. Yeah.
Yeah, LNAs.
Yep.
Are you using those?
No.
Because I believe if we do use those on the nanostring,
Yeah.
there's the prospects of developing an assay where you implement 12 primer pairs,
Yeah.
which are locked nucleic acids, that could potentially amplify hundreds of molecules.
Yeah.
And you turn it into a generic product that makes it a bit more appealing for someone like me that's using it on many different species, each species at a time.
Do you know, the thing is, I agree with you. There's the way NanoString have set this up is interesting. The only reason they built the elements chemistry originally was because people were asking them to build diagnostic assays. But if they did, they'd be providing all the analytes, right? And so the onus of performance falls back on NanoString every time. And the FDA wouldn't. This just becomes a mess, right?
Yeah, I think the locked nucleic acid chemistry and technology is now actually locked by Roche.
Yeah.
And that's
But the point is elements was made so that they could uncouple the NanoString elements from the oligos. So you just make oligos. But that kind of means you can play around, right? What frustrates me is I've never seen anybody play around. You could play around and order any variations and look at these crazy ideas, and you think people would. If I was in the lab, I would have. But you know, people just like to pay their money, get their result. They don't really play in the lab very much. But there's no reason that you couldn't, with some great ideas, do something really amazing with modified bases, for example. Yeah, 100%.
Just wonder how you design the general probe.
The design, what you do is you send NanoString there's a form you fill out. They don't charge for this at all. So you can have them design anything you want and not commit to anything, right? So you just tell them what you want to do. And it might mean that you specify ideally like a REF-seq ID or something for the gene targets. Or you can, yes. But if it occurs in a public database somewhere, anywhere on Earth, NanoString will find it and design an assay to it. If it's sequence that only you have, you can provide that sequence and they'll design to that. Because we've had examples of that. Like the New Zealand eels they're the only people who had that sequence. So that works. Or you can go in and say, I've got a problem. I've got these two bacterial species and I wanted to separate them. Can you design an assay that will separate them? And they'll go through and design an assay to do that. And then they'll report it back. So they've done all the design work in separating out a target set that would work. And they'll give it to you. You can confirm that, but they'll do a lot of this kind of bioinformatics, some pretty fancy bioinformatics. Other examples are people have said, I want to design, a probe set that will work in mouse and in human, or in all non-human primates, for example. And there's examples in xenograft situations where you want to detect the mouse and also the human variant of that gene. So there's some crazy things. You just specify all this and then they'll go away and design it. And they'll come back and they'll talk to you about it if they have any problems. So it's iterative. Yes. Yeah, they have their bioinformatics pipeline, which is, they call it the gauntlet because you got to run the gauntlet. There's gauntlet 2.0 now or something. It's really sophisticated, yeah. So you get all that for free. Yep.
Has anyone who says looking at mixed infections,
Yep. Yep. What's there? Yeah, there's been a lot of questions around that. They can design to anything, but bacterial species are kind of continuum, aren't they? They blend one into the other. People have wanted to do environmental screening to look at the health of soil and things like that, which is, we're looking at, you're probably best to use sequencing because you're not making any prior assumptions. This is a validation technology. You have to know your target, basically. But if you do know your targets, yeah, perfect. Yep. I don't know, actually. I know it turns up in funny places, but I couldn't answer that. Yeah.
By validation, RNA-seq, you love it as far as, yeah, it's a good initial exploratory, because that's what we.
Yeah. RNA-seq is a massive amount of data because you've got to sequence through the high expressors. So a lot of your sequencing ends up being junk because you don't learn anything. And to get into your low expressors, you've got to do an awful lot of work, a lot of reads. So it gets pretty expensive. I mean, costs are coming down, but you end up with, I don't know how many, what the data, it's terabytes or whatever it is now, but you got a lot of that to sort through, whereas this is just a really simple molecule count. Yeah. So if you know your target, yeah. 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 Well, there's still reverse transcription bias and there's still all kinds of biases in there, but the assays have come a long way over time and they're probably the best. RNA-seq's better than PCR, right? But there's still compromises in there. If you know your targets, I would do this. If you are in discovery mode, well, you really got to go RNA-seq and you can get pretty good. You can do, I'm not going to say you can't work with RNA-seq, you can. You just have to do the usual validations and you're right. When I gave you all those scary stories, that's not the end of the story. It doesn't mean it doesn't work, but a lot of people you saw make stupid mistakes and they publish things which are, should be retracted or binned. You don't do that. Well, we've gone right to the minute probably or just over. So that was a great session. I really want to thank you for coming to hear a technology talk. Technology talks are notoriously boring. I hope this one wasn't. But thanks for coming along. I hope this gives you some food for thought.